All right, well, let's welcome and salutations. Let's begin with some business. This is probably the most exciting part of the day for you. Everyone needs to remember to silence their cell phones. So if you don't have it in vibrate mode, we know you won't be able to resist looking at it, but at least have it in vibrate mode. And that goes for phones and other mobile devices. So if your iPad gets Verizon, that needs to be silenced as well. If you're not in The Universe is Expanding, reviewing the state of the art in stem cell derived cell therapy, you might be in the wrong room, but that's probably a good thing because I'm sure this is more exciting than whatever else you had going on this morning. As far as attendance, I have a note to remind you to not leave bags on chairs because it's going to be potentially a packed house. I'm not sure that prediction will necessarily come true, but if you see it's getting crowded, at least at that point be considerate and remove your bag. Otherwise, if you need a lot of shoulder room like I do, then I guess you can take 2 seats at this moment, just like an airplane with the middle row is untaken. As far as photographs and filming, if you are planning on preparing for next year's Oscars, it's unfortunately prohibited. You can't take a photograph and you can't film, and I guess somebody will be filming it, but it won't be you guys. And there are secret agents randomly distributed in the audience that you won't be able to identify, and they will take your phone away. So please honor that. Let's see. Also, for those of you that are planning on giving a talk later at this meeting, you need to check in at the speaker ready desk at least 4 hours in advance of your session, and if your session's in the morning, then that probably means the night before. And finally, the ASGCT has gotten rid of those pesky paper evaluations that get assaulted onto you as you're entering the room or leaving the room that you don't fill out anyway, but now you can do it on your app discreetly and do it right after the session. And you guys are probably more tech savvy than I am, but it says navigate to the session in the app, and then there's a clipboard icon, and then there's a series of questions. So please fill those out. Those are very important so that the meeting organizers know, who hit it out of the park. Today we'll have 3 Grand Slams, but in general, for those less exuberant talks, you might want to be able to put your recommendations. And then finally, the Wi-Fi is ASGCT 2018. And if you've been at previous meetings, I think they were probably 2017, 2016, et cetera. So that's it for your warnings. It's an exciting session today. The field is moving along. We're all excited that they're now approved adoptive cell therapies against leukemias and some lymphomas. But the price is still high, well worth it, of course. I was told to say that. But of course, there's great room in the field for bringing the cost down and also for accelerating the time to treatment. It still takes a long time to prepare these therapies. And if we could come up with a universal approach for doing that, that would be a great windfall for the field. There's a number of different approaches. You're going to hear about 3 of them today, whether it comes from stem cells or whether you can do it from allogeneic cells that are gene edited among the approaches. So our first speaker is Bob Valamehr. He's the VP of Cancer Immunotherapy of Fate Therapeutics. He has a long history of success in this area. He's been involved in modulating stem cell fate for quite a long time, and it's been a very fruitful career. He's giving us a talk today about the pluripotent cell-derived NK cells as a cornerstone approach for off-the-shelf cancer immunotherapy, and we're all looking forward to hearing that talk. So without further ado, Bob.
Well, thank you for that great introduction. It really warmed up the crowd, so that's exciting. And again, thank you for the invitation. And as mentioned today, I'll talk about how we're using iPSCs to create off-the-shelf cancer immunotherapy products. So forward-looking statement for your review. So obviously, we all know about CAR T cells. Pioneering work in CAR T cells in the early 2000s by Michel Satterline and others have really honed on the molecular strategy, on the targeting strategy, and in combination have resulted in clinical trials and approval of multiple products, as you see here, by Kite and Novartis. As you can see, the clinical trials are over 250 clinical trials today. I'm sure that's going higher. And so really, this rise in approval of CAR T therapy in such a short period has really paved the way for engineered cell therapeutics. And cancer immunotherapy is getting upgraded and needs a cellular platform. So as you go from a Model T to Formula One, which we've been hearing about, we really need a cellular platform. As kind of mentioned here, you can see how really being able to optimize CAR therapy as well as edit the cells themselves has really revolutionized the field. We're all here because we are editing cells. We are putting cells into patients. But as I mentioned, there needs to be a true off-the-shelf platform, advance the engineering technologies, and reduce the variability that's associated with cellular manufacturing. And also, where are the NK cells? We've been hearing a lot about CAR T and T cells in general, but we really haven't heard too much about NK cells. And these are very intelligent cells. They are multifaceted. Through their inhibitory and activating receptors, they can sense stress cells while leaving healthy cells alone. They don't have a TCR, so GVHD is not seen. They can activate the adaptive immune system by secreting chemokines and cytokines, and they have a CD16 Fc receptor to complement monoclonal antibodies through ADCC. So these cells are highly sophisticated and intelligent, but they also have been held back because of difficulties in genetic engineering, the lack of reproducibility, the donor-donor variability seen, and they have poor persistence. And so at Fate, our solution is to utilize iPSCs to create off-the-shelf hematopoietic cells. So we start off with a population of pluripotent cells, cells that have unlimited self-renewal capacity, as well as ability to differentiate into all 200 cell types. We uniquely take individual cells of iPSCs and put things in and out to create a library of different cell entities, and through clone selection, we can pick the cell that has the attributes that we want, while it does not have the attributes that we don't want, such as off-target gene editing. And so when we select the clone that we want, the one that is iPSC, that has the right modality, that can differentiate into the cell type of choice, we create a master cell bank. And that master cell bank can be used time and time to create the desired cells. As I mentioned, we focus on hematopoietic cells and NK and T cells thereafter. So in collaboration with multiple institutes that you see listed in the bottom, we are now creating off-the-shelf products that do not require patient sourcing, are consistent, reliable, they have the off-the-shelf potential, and they have unprecedented scalability. So that was a cartoon. Here's what actually happens under the microscope. We take any starting material, whether it's blood, fibroblasts, or other cell types, and we reprogram and engineer in a single event. We drop individual cells into a 96-well plate, and from there, as I mentioned, we screen for the modalities of choice. Once selected, we have a clonal bank. This population came from a single cell event. It's important to note that, and has the ability to basically expand as desired. So we can create large banks. And those banks, at the time of choosing, will be directed to become the cell type of choice. And here, as you can see, the shift has been made toward hematopoietic lineage. Obviously, all this we've been doing for 10 years, but it's been on the shoulders of pioneers in the field, as you can see here in pluripotent biology, hematopoiesis, and oncology. So as you can see here, we drop a single cell into a 96-well plate. After 24 hours, that cell population begins to double and eventually expands into the homogeneous population you can see here. Through small molecule guided culture, we maintain the iPSCs in a pure state. As you can see here, the population is pure for SSC and TROP181, surrogate markers of iPSCs. as well as genomic stability. Because the cells are maintained in a happy state, they can be dissociated as single cells, they can be expanded feeder-free, and maintain their attributes. So these cells are IPS Cs, these cells are genomically stable. Having that platform really enables us to do precise engineering. So as you can see in the bottom, on the left-hand side, we have a parental population of iPSCs that does not express GFP, as expected. We targeted a safe harbor locus with homology arms and put in 3 genes into the targeted safe harbor locus. On the right-hand side, our strategy was different. We took a population of iPSCs that is expressing HLA class 1, and as well as beta-2 microglobulin, and went to target the beta-2 microglobulin, a biallelic disruption, to pick the individual cell that has now the loss of B2M. And those 2 strategies yield in pure population. So on the left bottom, you can see that now we have a pure population, a single cell-derived population that contains the cassette of choice. And on the right-hand side, we have now a population that's pure for HLA class 1 loss because B2M is no longer expressing. The second component of this platform is to be able to differentiate in a very efficient, reproducible manner the cell type of choice. So here, in a 10-day process, we can take iPSCs and convert them to a CD34 positive population, which then subsequently is differentiated into NK cells. As you can see here, in a stage-specific manner, we go from a population that's 34 positive to a population that's pure for CD56, as well as expressing other markers of NK cells, such as NKP30. This process has now been tech transferred to University of Minnesota GMP Facility, and as you can see here, it's now scaled at 5-liter G-Rex capacity, so we culture multiple 5-liter G-Rexes. We create dose banks of basically here pure population of CD56, CD45, and the cell population lacks CD3 T cells. So this process, because it's stage specific and it goes from iPSC to CD34 to NK cells, is highly proliferative. 1 iPSC gives rise to 1 million NK cells. And as we'll talk later, this scalability satisfies your entire clinical trial. And so as I mentioned again, we just basically run the campaign over and over again because we start from the same starting material. So obviously, this is an iPSC starting population, so we need to make sure that there are no iPSC residuals in the final product. And as you can see here, either with flow cytometry or qPCR, we do not detect iPSCs in the final 44 days. So the 44 days consist of 2 different enrichment strategies. The first one is a mechanical strategy where we enrich for CD34s, and the second is a differentiated enrichment strategy where the cells naturally expand the NK population. So here, as I mentioned, up to a resolution of 1 in 2 million, we have to date not identified any iPSCs in our NK product that's being made at University of Minnesota. Gene expression profile, as expected, maybe not as expected, but as we were hoping for, falls basically on top of primary NK cells. So our iNKs cluster well with primary NKs and away from other iPSC-derived products or starting material. And so another key concern was when you make NK cells, there's a phenotypic way you can profile, there's a gene expression way you can profile, but NK cells, as I mentioned earlier, have the inherent ability to sense stress cells and target them while leaving healthy tissue alone. And so this was kind of our way to make sure that we really have a truly mature NK cells. We basically did the study where we co-cultured our NK cells with K562, so these are HLA class 1 null leukemia-derived cell lines, and the NK cells, whether it's iNK or whether it's primary NK, they love to attack these. And as you can see, as you increase the ET ratio in orange, the specific killing goes up. That's expected. Everybody sees that. What was exciting to see was in purple, when we combined the same NK product with PBMCs from patient or healthy donor derived, we didn't see any increase in specific killing as we went up in the ET ratio. So that was exciting to see. And in turquoise, there wasn't any synergistic effect when we combined all 3. So this really made us comfortable that we were making a product that is very close to its natural form. And another key important point that we had to overcome was the ability to have a product that is thawed and directly infused into the patient. In order to facilitate off-the-shelf strategy, you need to make sure that you can characterize a product up front and deliver subsequently and have the site be able to, with minimal manipulation, treat the patient. And so what we did was, through optimization strategies, we were able to create a cryopreservation method where our cells coming directly out of thaw are still highly effective. So as you can see here in blue, when expanded primary-derived NK cells are immediately thawed, they have lost their, to some degree, have lost their ability to target the tumor cells. As you can see on the y-axis, as you go down, target cells are eliminated. You do need an overnight recovery to have these expanded NK cells be able to function optimally, as you can see there with the dotted blue line. But I hope you can appreciate here that whether it was immediately defrosted or whether it was overnight recovered, the killing curves look very similar. And in fact, this is how we do most of our studies of the final thawed product. Which leads me to this kind of complicated slide, but I'll basically have you focus on these two columns in blue and red, where we either took, we took an ovarian cancer cell line and basically treated the mouse 3 times with a frozen product. So this is thawed and directly infused into the mouse. As you can see with the schema, they're treated on days 4, 7, and 10. And as you can see here, on day 38, there are many mice with the treated off-the-shelf INK product that have controlled the tumor burden, unlike the control. And when you look at the significance here, there is significance between the tumor only as compared to the off-the-shelf product. So that was exciting, and that has kind of led to some of the biological analysis. What is it that really is making some of our NK cells better than the primary derived NKs? And 2 of the things we've identified. One is that obviously they have longer telomere lengths because they come from IPS Cs, so potentially they have more rejuvenated capacity. And second is we picked clones that were able to produce more granzyme per cell. So not only these cells are rejuvenated, they actually have more killing capacity. So this is we're starting to understand some of the biology here behind our improved process and cells. All that has kind of led us to this unique pipeline. I think a lot of various platforms have a unique modality that they target many different antigens. Here we have a cell platform, and as you can see, we have off-the-shelf NKs and off-the-shelf T cells. For off-the-shelf NKs, we have a variety of different cell types, whether it's FT500, which is an allogeneic NK cell, non-engineered to be combined with checkpoint inhibitors, and I'll give a couple of slides on how that makes sense. And then from there, we have engineered cells to put in modalities to improve targeting and killing of tumor cells. And in the bottom are off-the-shelf T cells, which is in collaboration with Memorial Sloan Kettering under the leadership of Michelle Satterline. I will not talk about that right now because I believe Michelle will talk about it in the subsequent presentation, but that is also where we're very excited, making off-the-shelf T cells. And as you could imagine, there's a lot of potential there as well. So NK cells in combination with checkpoint inhibitors. As we all know, NK cells secrete a lot of interferon gamma and TNF alpha when activated. So as you can see here in orange, when NK cells are basically co-cultured with target cells, you see an upregulation of interferon gamma. We took the culture, the conditioned media from there, and we added it to T cells. So whether the T cells had fresh media or whether the T cells got the supernatant from activated NK cells, as you can see on the right-hand side, we looked for CD69, an expression or a surface marker of activation of T cells. As you can see here, with 4 hours and 24 hours, we see an upregulation of CD69. So this is most likely, this is common to most people here. They know that obviously when you take NK cells and co-culture them with T cells, you get an activation. So the soup actually is what, from this study, elicits the CD69 response. Secondly, we wanted to see if there is a synergistic activity between NK and T cells. So what we did is we created this spheroid assay, and created aggregates of SCOV3, which is an ovarian cancer cell line, and then basically mixed the aggregate with either T cells or a combination of T and iNKs. And as you can see here, either with the image or with the killing curve, the combination of T cells with iNK basically eliminated the entire aggregate. So this shows that basically these two cell types work in synergy in this one assay. And finally, we want to see if the iNKs can elicit some sort of recruitment of the T cells. So in red, we basically put iNKs IP into the mouse, and as you can see, we didn't see T cells in the blood or in the peritoneal cavity, which is expected. No T cells were introduced into these NSG mice. In purple, we added the T cells IV, and of course we saw it in the blood and didn't see much in the peritoneal cavity. However, when we put the iNKs IP and the T cells IV, we saw a nice presence of T cells in the peritoneal cavity. They left the blood, they went to the peritoneum. So that was exciting to see. So these studies have really justify our first allogeneic strategy, where we will combine the iNKs and with checkpoint inhibitors, and this IND is in the process of completion, and we hope to have an update for you by summertime, and hopefully the first patient treated in fall. So how about engineering? So as I mentioned, NK cells have been held back in part because of inability to engineer. And so what we did is we, through the work of University of Minnesota, we have a novel transgene for CD16. It has 2-point mutations. One elicits a high affinity to the FC receptor, or FC through the FC receptor, and the second is it prevents cleavage. So CD16 is cleaved upon activation. We took this transgene, put into iPSCs, and created a CD16 iNK, which we call FT516. And this is a very unique strategy that is amenable to multiple different clinical indications, whether monotherapy or in combination with monoclonal antibodies, engagers, CARs, or checkpoint inhibitors. This highly tuned CD16 bearing NK cell, it's an ideal cell type for all these different strategies. And so again, because we use an IPS C and we grab a single cell, here you can see the clone expresses CD16 and is an IPS C. But as I mentioned earlier, we can easily take that IPS C product, make it into NK cells, as you can see. On the right-hand side, by the flow plot, this population is pure for CD56 and has CD16. And as I mentioned earlier, because of our highly tuned scalable process, we can take 1 million iPSC, which is a small Petri dish, and differentiate that into 1 E12 NK cells, which is enough for your first clinical trial. And so we wanted to start investigating the function of the CD16, and as you can see in the top, primary derived NK cells express CD16 in culture. As you can see, there's a homeostasis of CD16 expression on the far left panel, and there's no CD107A because the cells have not been activated. But once activated with target cells, you see a shift in CD16 expression because CD16 is being shed, and you see an upregulation of CD107A. Same thing happens with PMA eomycin. Here you can see that the pure population of CD16 of the iPSCs, when activated, does not elicit any shedding of CD16. The CD16 remains at 100% of the population. There is a CD107A upregulation, but CD16 remains to be highly expressed. Same thing is observed when PMA eomycin is introduced to the cells. And so regardless of what you do, as you can see in orange, the percent CD16 of the population remains nearly 100%. So that's great, but how about the biology? And so basically, we know that CD16 activates ITAM and subsequently phospho-ZAP70 and phospho-SYK. And so as you can see, a bunch of different controls, regardless of whether you have primary NK or CD16 iNKs, there is no upregulation of phospho-ZAP70. But when you introduce the anti-CD16 with a secondary antibody, you can see that CD16 elicits a stronger phospho-ZAP response. And so we see the molecular design work, we see the cell signaling work, but how about function? And so we went head-to-head in a long-term killing assay of about 70 hours, and we compared cord blood, peripheral blood-derived NK cells, against FT516. And as you can see here, worth mentioning, we targeted A549, which is a lung carcinoma cell, which has low expression of HER2 and high expression of EGFR. And so two things to see here. One is you see a very enhanced ADCC effect, as you can see with anti-EGFR in the purple line with FT516. Because of the CD16 and because of the clone selection, you get augmented killing. That killing is actually controlled with through the antibody, and if you use an antibody that is not as binding here, Herceptin, because these cells are HER2 low, you do see a nicely controlled strategy. That anti-HER2 curve is same as with or without antibody. So the cells have an inherent capacity to kill and are augmented through ADCC. Same thing is seen with other cell lines, or target cell lines. And so obviously we want to go in vivo. We wanted to use our multi-dosing strategy because we can. This is an off-the-shelf product. And so we took basically Raji cells that are CD20 positive, combined it with Rituxan, which is an anti-CD20 antibody, and did a multi-dose strategy, days 4, 11, 18, and 25, comparing ex vivo expanded, using either ex vivo expanded NK cells for the multi-dose strategy or FT516. Again, a busy slide, but if I could have you focus on these two columns. Ex vivo expanded NK cells in combination with Rituxan did control the tumor burden to a degree as compared to the untreated or the antibody alone. However, FT516 in combination with Rituxan dosed 4 times really showed impressive control of the tumor burden, at least in 3 out of 5 mice, as you can see here in day 28. And when increased, we see that time over time, about half the mice, as you can see in black, which is FT516 with Rituxan, at least half the mice are maintained in long-term survival. Where in purple is the ex vivo expanded primary derived NK cells with Rituxan. Basically, most of the mice are seen dead by day 73. The control is day 49. And so FT516 has really unprecedented ability to be characterized and to be safe because it is made it as a one-time event. It can be characterized as a one-time event. So FT516, it is single cell derived, we know that. Very uniquely, we know how many copy number it has. We know where the FT5 or the CD16 has integrated into. We know that the product is stable. We've done multiple tests on it. So this really becomes, for the first time, an engineered product that before treating patients, you know exactly what the properties are, and every patient will get the same product. And so we hope to file the IND on this product in the second half of this year, probably toward the end of this year. And so because we can genetically engineer these cells, we want to go on to the next challenge. So we put in CD16, and we showed that it works. But how about knocking something out to improve function? And so Daraa, Darzalex, is an anti-cancer drug that binds to CD38 and is an ideal antibody for multiple myeloma. As you can see in the orange on the left-hand side, Daraa helps binds to CD38 positive tumor cells, bringing in NK cells to and elicits ADCC. However, the same mode of action creates fratricide, where basically the antibodies the Daraa binds to NK cells, and other NK cells come in and kill it. And so you have this unfortunate strategy where a potentially amazing product is held back because of the NK cell, lack of NK cells in the patient. And so what we wanted to do is we wanted to see if we can knock out CD38 and answer 2 questions. Can we go from an IPS C to a CD38, or IPS C to an NK cell with CD38 loss? And once we're there, can these cells maintain viability in the presence of Daraa? And so because we are dealing with the same population, it's very easy to do gene editing because you have that population. When you test the specific combination of guide and Cas9, you go back to the same population. It's not that you tested it on something and you have to go test and actually apply it to something else. So here we went through our screening of guides and identified those that had the best cleavage percentages, and basically, through an unprecedented strategy, looked at 28 different clones and identified the one that we wanted. So basically, we had some clones here. One had no, maintained the biallelic integrity of CD38. We didn't see any clones that would have basically one allele disruption, and we saw multiple clones that had biallelic disruption. So whether the biallelic disruption was different or was the same in each allele, we picked the one that we wanted, and basically ended up with a product that is now dubbed here FT538, that is highly enriched in CD16, but does not express CD38. And so first, we want to make sure ADCC function was maintained, because CD38 can play a role in binding and recognition. And so as you can see here, FT538, when combined with either Dowdy cells or RPMI 8226, you can see that ADCC is elicited by the combination with either Rituxan or Daraa, as you can see with TNF-alpha and CD107A expression. And so next was we wanted to see if there's fratricide. So we basically took the target cells out and just cultured NK cells. And we took the wild-type NK cells and either cultured them with anti-CD20 or anti-CD38. And we saw in its linear culture, we saw that NK cells with Daraa did elicit an upregulation of TNF-alpha and CD107A. We did not see that with 538, which has CD38 loss. So this is exciting to see, because essentially the genetic engineering led to an improved function. And when you look at viability, you can see that viability is maintained with FT538 in the presence of Daraa. So last few slides. As I mentioned earlier, persistence is an issue with NK cells, so we wanted to basically make the cell autonomous. We wanted to make sure that the cells can grow on their own, so we added a membrane-bound IL-15, because whether it's in culture, whether it's in the mouse or in the patient, you have to give systemic IL-2 or IL-15. If you give IL-2, Tregs come up, so this becomes a problem. And obviously, through pharmacodynamics, it's hard to get the cytokine to the NK cells. So here, we want to make the cells autonomous. So we targeted IL-15, created a membrane-bound IL-15, put it into the IPSCs, tagged with LNGFR. You can see that the cells express IL-15 in a uniform manner, and when they're CD56, you can see that the population is pure for 56 and LNGFR. And real quickly, you can see that now we have a cell autonomous product. So when we put these engineered iNKs into culture without cytokine, you do see an autonomous activity, as you can see in red, for at least about the first 6 days, and then that drops. But which is good, because we don't want a long-term persistent product. We have the ability to multi-dose, so we will basically utilize this strategy. And in vivo, you can see the same thing. Only in mice that were not treated with IL-2 or IL-15, only the membrane-bound iNKs were able to show persistence over a 3-week period. And so finally, last 2 slides, I hope I've proven to you that iPSCs are the ideal platform for cell engineering. They are renewable, they can be single-cell engineered, and they have the propensity to differentiate into all 200 cell types. That single-cell-derived master cell line can be banked, can be made into working cell lines, or working cell banks, and can be differentiated into T and NK cells, basically reach every patient in need. And so finally, as acknowledgements, I want to obviously thank the people at Fate Therapeutics, CERN for funding FT516, and obviously the people you see here who have basically made all this come true. Without them, none of this would be possible. Thank you.
Well, thanks for a great talk. That was really interesting. Due to the time, we're going to save most of the questions for the panel discussion at the end, but if there's 1 or 2 burning questions and somebody doesn't feel like they're going to be able to stick around, I'm willing to entertain that. Otherwise, I think we'll move on. Is that all right with you, Bob?
Absolutely.
Okay. All right. And we'll bring you back.